So now there's actually two other ways in which apoptosis can be initiated. It doesn't only have to be initiated by p53. It can also be initiated extracellularly through signals. So it can either be initiated through the secretory route or the non-secretory route. So I'll discuss both of those here. With the secretory route, you have the natural killer cells and cytotoxic T lymphocytes or cytotoxic T cells, and they will recognize the cell as being, they'll recognize signals on the cell, bind, and release perforins and also granulocytes. Okay. Perforins will form a complex similar to like the MAC complex that actually gets integrated into the membrane, and it allows for an influx of calcium. And the textbook doesn't actually explain what the calcium does. The granulocytes will actually be secreted as vesicles, which are endocytosed into the cell. And within the cell, they actually have perforin as well in the vesicles, which will combine and release granulocytes out of these vesicles, and these can go on to cleave the caspases. So for example, procaspase 3 can get cleaved to become caspase 3 initiating apoptosis. So this is the secretory route because these cells are actually secreting the proteins that initiate apoptosis. Now, in the non-secretory route, you actually have cytokines that are released. So I'm just going to draw up, there's two kinds of receptors. There's the FAS receptor, and then there's the tumor necrosis factor receptor. Okay, so FAS ligand can come and bind to FASR, and tumor necrosis factor can go and bind to the tumor necrosis factor receptor. And these actually, these are transmembrane proteins or receptors that go on to activate. There's two proteins that are bound to them. There's the FAS-associated death domain and the tumor necrosis factor receptor associated death domains. And what these do is these actually cleave, there's procaspase 8. They cleave procaspase 8, and they'll do a lot of them, right? A lot of these procaspase 8s. And these procaspase 8s can go on to form a complex that are then catalytically active to cleave procaspase 3 again, and which forms caspase 3. Okay, this is actually called, like, this is through type 1 activation. There's another way in which they can become activated. If there's not enough procaspase that is cleaved to actually form this complex, what happens is caspase 8, once it's activated, can go on to cleave BH3 only activating protein. And as discussed before, this BH3 activating protein then activates BAC and BAX, which goes and forms a pore in the mitochondria, and then it goes through the same steps as the intracellular method of initiating apoptosis. One more thing that I'd like to add is so once apoptosis, once it's initiated and the caspases start to degrade the cell, they degrade like cytoskeleton, nucleases, all this stuff, and allow for the formation of apoptotic bodies, right? And these apoptotic bodies They all have the degraded substance in them. Okay.